today I'll talk about learning from data in single-cell transcriptomics. So I'll give a little bit more background and introductory comments before I get to lineage inference and trait-seq. I've had a wonderful time working on this topic with current and former PhD students and postdocs, in particular Philippe Boileau, Nima Hejazi, Fanny Perraudot, David Erisso, Hector Roux de Bézieux, and Kelly Street, whom you know already, as well as Koen Vandenberg. Special thanks to John Nye, my collaborator of 20 years, for motivating most of my work in this area. He's been absolutely terrific to work with. And I'd also like to thank my collaborators in statistics and computer science, in particular, Lieven Clément, Elizabeth Purdom, Jean-Philippe Vert, and Nir Yosef. And here's an outline of my presentation. Basically, it's going to be a survey of the methods that we've developed throughout a single-cell RNA-seq workflow to address questions related to the differentiation of stem cells. So we'll start with a few words about the biological question that motivated our work. Then we'll move to exploratory data analysis, normalization, expression quantitation, a few words about cluster analysis, and then the rest of the talk will be devoted to lineage inference and the study of differential expression on trajectories. So a few words about high throughput sequencing. As you know, microarray and high throughput sequencing technologies have revolutionized how we do biology by allowing us to study biological processes on a genomic scale. And most recently, single-cell transcriptome sequencing allows us to measure the expression levels of an entire genome at the resolution of single cells. And the ability to work at the resolution of single cell is very important to study a variety of biological processes, like how stem cells differentiate, or also to help us discover new cellular subtypes in brain tissues. So RNA-seq studies provide a really good example of the range of questions we encounter in a data science investigation, where the data are complex in a variety of ways. They're complex in terms of their dimension. We can have either the small and large p setting or large and large p setting that we have now with single-cell RNA-seq. There are various levels of processing with the data. We could be working with raw images or with already normalized matrices of expression counts. The data are of various types. They could be image data, string data, integer matrices, or graphs. We're not limited to the experimental data, but we have metadata. In other words, we have data, like data on the genes in the form of ontologies, or data on the cell in the form of quality control variables or variables that reflect the experimental design. The data are sparse, particularly in single-cell RNA-seq, where we have zero inflation. There are large technical biases related to batch effects, but also sequence composition effects. And we're often faced with a problem of low signal to noise. So before we get into the specifics of the biological question, I'll summarize the main steps in a data science workflow in general. And that's because this is essentially the process that we followed when collaborating with the Nye lab. So it all begins with a driving biological question, in our case, the study of stem cell differentiation. Then a very important step that's far from trivial is to frame and translate a possibly vague biological question into a question about data and about statistics. So very narrowly speaking, a question in statistics would be identifying a parameter of interest that we want to estimate or test hypotheses about. Then comes the design of the experiment or study to make sure that we can actually answer the question of interest, and also that we answer this question as accurately as possible given the resources that are available to us. Then we collect, store, and access data. A very important step in single-cell RNA-seq data analysis is exploratory data analysis. And that's very important for quality control purposes, and also to get a sense of the main features of our data, whether they're good or bad features. So I cannot emphasize enough the importance of looking at data. Otherwise, you risk having garbage in, garbage out. If your data are flawed from the beginning, there's no way you can get reliable biological conclusions from them. It's also very important to use the right types of graphical summaries. There's a huge toolbox of data visualization methods, and it's good to go beyond simple histograms and pie charts. Then comes pre-processing of data, in particular normalizations, which I'll mention. And this seemingly mundane and often uninteresting step can actually have a huge impact on the results of downstream analysis, often a larger impact than the choice of machine learning methods. So it's really important to get normalization right. And here in 7, statistical inference of machine learning is the typical entry point or comfort zone of statisticians and computer scientists. This is where our data can be represented in the standard format of a matrix n by p. So this is an important step, but it's important to recognize that it's only one small aspect of an entire data science pipeline. And in order to do this step correctly, it's really important to understand the biological context for our data and also the steps that came upstream of the statistical inference or machine learning. And it's also very important to think about the interpretation and validation of our results and how to translate them back into meaningful biological insight. And then finally, this leads off into whole new domain questions. So, whoops, I'm just going the wrong way here. So data science is a highly iterative process where we go back and forth between the different steps that I've mentioned above to refine the question, refine the study design, refine the analysis and the validation. It's also highly collaborative and interdisciplinary. It's really crucial to have feedback from the domain experts throughout the process. Statistical software is also key for implementing all the steps of the workflow. And finally, a notion that's getting traction after years and years is the notion of computational reproducibility. And that simply refers to the ability to reproduce the results of a study given the methods, the data, and the code. And why is this important? Well, it's just good scientific practice. It's part of the scientific method. And I want to make sure that we distinguish between this rather narrow notion of computational reproducibility and the much more ambitious notion of biological reproducibility of the results. So whether the results of one study hold in a brand new study. And I usually like to quote John Tukey, who was a data scientist before it became popular. So this is a quote from about 50 years ago. Far better an approximate answer to the right question, which is often vague, than an exact answer to the wrong question, which can always be made precise. Okay, now a few words about the biological question. So the work that I will present was motivated by our collaboration with the Nye Lab, with John Nye's group, who is a neurobiologist. And he's interested, him and his group are interested in studying how stem cells differentiate in the mouse olfactory epithelium. This work has potentially very important application, in particular regarding the development of treatments and therapeutics to ameliorate neural tissue damage and degeneration. They're focusing on a very specific type of stem cell, the horizontal basal cell, or HBC. That's an adult tissue stem cell. And they're studying how HBCs differentiate by using the p63 protein as a molecular switch. In normal condition, the HBCs self-renew and p63 blocks differentiation. But when p63 is downregulated or knocked out, this is when the HBCs start differentiating. So in order to investigate how HBCs differentiate, they perform single cell transcriptome sequencing on both wild type and p63 knockout mice. So here's a diagram showing the biological system that they're studying. It's the mouse olfactory epithelium, which is located here in the mouse's head. And here's a diagram showing the different types of cells that we'll be monitoring in our study. So you have, would be the outside of the epithelium. You have the neurons here on top, the HBCs here in green, the global basal cells here in dark blue, and these sustentacular cells at the top. So we're going to focus how these green cells, the HBCs, either self-renew or differentiate into a variety of cells. In particular, they can differentiate into sustentacular cells, as well as neurons and microvillus cells. So we'll follow how these cells differentiate and what are the genes involved in these differentiation mechanisms. So our point of entry is already a little far in the data analysis process, in the sense that we won't be performing image analysis, we won't be performing read mapping. We'll take these steps as given, and we'll start with matrices of expression counts, where the rows correspond to cells and the columns correspond to genes, and the count is the number of reads that maps to a particular gene region. So that's a proxy for the expression level of that gene. So these matrices come after a bunch of steps that we take for granted. And the types of questions that we'll want to address with these data are what are the different types of cells that we have? So that's a cluster analysis questions. What are the differentiation paths for these cells? That's the trajectory inference question. And also, what are the genes that are implicated in these differentiation processes? And that's what we're going to be discussing with the new trait-seq method that Yang referred to. Any questions so far? Okay, so here's a diagram of the experimental design. So it's a bar plot where each bar corresponds to a batch, and the batches are colored by the biological conditions. So we have the different time points after treatments for the knockout here, different colors, and we have the wild type. And the height of a bar represents the number of cells in that batch and biological condition combination. What you can see from this plot is that batch effects are nested within biological effects, but we'll still be able to detangle the biology from that. So here are the main steps in the workflow that we've developed to address a range of questions in the analysis of these single-cell RNA-seq data. And in gray, I've indicated the R software packages that we've developed to implement the methodology for each of the steps. And we've released these packages through the Bioconductor project. So exploratory analysis, I'll mention a few words about that. Normalization. I won't have time to discuss dimensional reduction, but one of our group members, Philippe Boileau, developed a method for sparse contrastive principal component analysis. It's a way to do dimensional reduction and at the same time remove unwanted variation and produce sparse principal components. We have a method, a framework for expression quantitation based on a zero-inflated negative binomial based model, and that handles zero inflation and over dispersion, and it can be used for a variety of purposes, like adjusting for known and unknown factors of unwanted variation. So that's normalization, dimensional reduction, and it provides weights that you can use in differential expression analysis. And I won't discuss that either now. I'll mention only briefly cluster analysis, and then the bulk of the talk will be devoted to trajectory inference. And I'm going very quickly through these slides, and if you're interested, I'd be happy to provide you the slides so that you can go through them at your own leisure. So let's look at data now. So first, we'll look at the phenomenon of zero inflation. So this is a bar plot of the proportion of genes with zero count per cell. And here, so you have one bar for each cell, and the bars are color-coded according to the batch. So what you can see is that there's a huge amount of zero inflation. Over 80% of the genes can have zero count in a particular cell. So this is very severe sparsity and something we need to address. So we do some filtering already of the cells based on the proportion of zeros that they have. These are box plot of QC measures that we have at the level of each sample. So these QC measures reflect alignment properties of the reads as well as sequence composition of the reads. So these are one box plots for each batch, and these are different quality control measures. So the take-home message from these box plots is that the quality of the sample varies from batch to batch and also within batch. So something to think about when we do normalization. More importantly, there's an association of the read counts with the sample level QC measure. And this can be seen quite strikingly here in this scatter plot of the first principal component of the QC measures and the first principal component of the read counts. So here you have a dot per sample, and they've been color-coded by batch as well. So here you have a negative association in this particular example between the QC measures and the count principal components. So something to account for when we do normalization. So now let's look at the read count at the level of genes. So these are box plots of the log counts. You have one box plot, they're very narrow because we have a lot of cells. So you have one box plot per cell, and the box plots are colored according to batch. And these are similar box plot, except instead of computing the log counts of the genes, we compute their relative log expression. And the relative log expression is the log ratio of read count to the median read count across cells. It's more informative to look at the RLEs because if the distributions of read counts across cells are similar, the RLEs would be centered around 0 and evenly spread around 0. And here you can see that's far from the case. There's a huge variation in read count distributions, both within and between batch. So something to account for in normalization again, because within a batch, you would only have, there's no biological variation, it's only technical variation. So this brings me to general remarks about normalization. First, what is normalization? Well, the goal of normalization is to adjust read counts for either gene level or sample level unwanted technical effects in order to allow meaningful comparisons of expression measures either between genes or between samples. And normalization is essential before you do any downstream analysis, whether it's clustering or differential expression analysis, because we want to make sure that when we see differences in expression measures, these are truly due to the biological effects of interest and not unwanted technical artifacts. And normalization is even more important for single-cell RNA-seq than bulk RNA-seq because we have increased levels of technical noise and zero inflation. So before we dive more into normalization, just two key questions. So first of all, does it matter? Before we start studying this and trying a bunch of methods, does it make a difference? It turns out yes, emphatically yes. The choice of normalization method can have a greater impact on the result than the choice of downstream method for differential expression or for clustering. So it's important to get that step right. Then the next natural question is, given that it matters, how do we choose a normalization method? And that's far from obvious. The proper normalization is data set dependent. And in the absence of control samples or genes, it's pretty hard to select a suitable normalization procedure. So we've attempted to address this question using the SCONE framework. So SCONE does a bunch of things. It implements a range of normalization methods, simple global scaling methods, as well as quantile-based methods and regression-based methods. And it also computes a range of normalization performance metrics that focus on three properties of the data: the clustering of samples according to factors of wanted and unwanted variation, the association of expression measures with factors of wanted and unwanted variation, and also the between sample distribution of expression measures. And SCOAN provides numerical and graphical summaries of the read counts and of their metrics to help you decide on the right normalization procedure for your data. So here's a figure showing the type of diagnostic plots that the SCOAN package gives you. And I'll show you a few in greater detail now. There's a shiny app for SCOAN that can help you navigate through the workflow. So we applied SCOAN to our olfactory epithelium P63 data set, and we looked at 172 different normalization procedures. So we looked at a bunch of global scaling methods like TMM, DSeq, or no scaling at all. We applied RUV, so this is a factor analysis method. We also adjusted for biology and batch or not using a regression-based model. And this is one of the main diagnostic plots that's provided by SCOAN. It's a biplot of the performance score. So it's a little, I'll spend a bit of time describing it. Each point in this scatter plot corresponds to one of the 72 normalization methods. And for each method, we've computed 8 metrics by SCOAN. And to reduce dimension from 8 to 2, we've done principal component analysis. And we've colored the points corresponding to each normalization method based on an overall performance score. So in yellow, we have a good normalization method. In dark blue, we have a bad normalization method. So for our P63 data set, SCOAN selected among the good methods, a method that does full quantile normalization and that normalizes for quality control using the first 4 principal components of the quality control metrics and adjust for biology. Another good method was full quantile normalization and adjustment for the first 2 principal component of QC measures. They perform quite similarly. As you can tell here, there's a cluster of yellow points corresponding to good methods. So let's look at the data now after normalization. So these are our box plots of relative log expressions after normalization. So they're not quite centered around 0, but definitely more stable in terms of their width compared to the plots that I showed you earlier here. So normalization did what it's supposed to do, which is make the distributions of read counts more similar between samples within a batch and also between batches. How did it do in terms of the association between read counts and sample level QC measure? Well, because we adjusted for QC measure as expected, it decreased the correlation between QC measures and read counts as shown by this plot. So it did what it was supposed to do. And I could talk way more about normalization, but I'd like to get to the rest of the topics in the talk. So I'd be happy to discuss this further offline or point you to some of our other results. So a few words about cluster analysis. So cluster analysis of samples or genes is a very common task in RNA-seq for the purpose of discovering novel classes of genes or cells, but also for exploratory data analysis. As you know, cluster analysis is a difficult task because no matter what the data or clustering algorithm, we'll get clusters. Clustering results are also sensitive to the data and to the choice of algorithm and tuning parameters. And in practice, we often try many clustering algorithms before settling on an appropriate one for our data. Also, there is a trade-off between the cluster resolution and the replicability of the clusters across data set. In general, the more cluster we have, the less stable they are, the less replicable they are across data sets. And finally, clustering results are very hard to validate when we don't have controls or ground truth. So you may ask, why again, why would I want to develop another clustering method? Well, this is a good question. This is not what we did. We did not develop a new clustering algorithm. Instead, we did 2 things. In our seq, we provided a general and flexible framework that enables researchers to apply and compare a range of different clustering algorithm and tuning parameters. And from this range of clustering result, it generates a stable consensus clustering. We also developed in Dune a cluster merging procedure to help navigate the trade-off between cluster resolution and replicability across data sets. So I'll just skip this and then go straight to trajectory inference in the interest of time. But our trajectory inference work is based on having clusters from RSEC, which we applied to our P63 data set. Okay, so this really brings me to the heart of the talk and the main goal of this experiment that our collaborator John Nye did with the mouse olfactory system. So again, if you remember, they were interested in studying how HBCs could differentiate in the mouse olfactory epithelium, and they used this P63 protein to trigger differentiation. Without the knockout of P63, the cells would keep self-renewing. So to address the inference of lineages when these HBCs differentiate, we developed Slingshot as a flexible and robust framework for inferring the cell lineages and also pseudotimes for where the cells fit on each lineage. And this is the work of Kelly Street that's here present at the talk. So Slingshot allows the identification of any number of novel lineages. So not just two lineages, it could be any number of lineages. And it also allows some supervision to incorporate subject matter knowledge. For example, if we know that there are certain cell types that are the terminal states in the trajectories, we can feed that knowledge to Slingshot and help it do a better job with the rest of the trajectory. So there are two main steps in Slingshot. The first step is the inference of the global lineage structure. In other words, the number of lineages that we have and where they branch. And to do this, we use a cluster-based minimum spanning tree. And the second step is the inference of the cell pseudotimes along each of the lineages. And for this, Kelly developed a method known as simultaneous principal curves. And the Slingshot method is implemented in this R package, Slingshot, released through the Bioconductor project. So in terms of input-output for Slingshot, the input is a reduced dimensional representation of our normalized gene expression measures and a clustering of the cells. So for the clustering of the cells, we use the results from RSEC applied to the OE data set. And when we applied our resampling-based sequential ensemble clustering method to the OE data set, we got 6 major groups of cells. The HBCs, of course, the globose basal cells, microvillus cells, immediate neuronal precursors, immature and mature olfactory sensory neurons, and immature and mature sustentacular cells. And these groups of cells or cell clusters correspond to a variety of states in the differentiation processes of stem cells into neurons and sustentacular cells. So we feed these clusters to Slingshot along with a dimensionally reduction of the data. And what Slingshot outputs are the cell lineages. So these are essentially ordered subsets of cell clusters. And it also outputs cell pseudotimes for each of the cells, as well as weights for assigning the cells to each lineage. So we don't force a cell to be in one lineage only. Cells can have weights for belonging to different lineages. So for dimensionally reduction, we have found principal component analysis and ZNB-Wave to work quite well, but you could also use a different dimensionally reduction upstream of Slingshot. We like to work with cell clusters and implement a minimal spanning tree over the cell clusters because it tends to be more stable than working with cells only. And we have some semi-supervision of Slingshot by specifying root nodes and leaf nodes. And there's also an ability to define an outlying cluster, which we call the omega cluster. Once we have the skeleton of the trajectory, then we infer the pseudotimes and the weights using simultaneous principal curves. This is an extension of the principal curves methods proposed by Hastie and Stutzel in '89. When we have branching lineages, we further perform a shrinkage step to shift the curve towards a consistent path where there's more shared cells in common. But then we allow the curves to diverge later on in the path. And then we obtain the pseudotimes by orthogonal projection onto the curves. So here are the results of applying Slingshot to our olfactory epithelium data. So again, for the cell cluster, we used the RSEC results. And we also did some supervision of Slingshot by providing to it known terminal states. So we knew that we had mature sustentacular cells, microvillus cells, and mature olfactory sensory neurons as our terminal states. So we provided that knowledge to Slingshot. It turns out that only providing the first type of cell had an effect. For the other one, Slingshot was still able to infer the correct trajectory structure. So Slingshot identified 3 lineages. We had one lineage from HBCs to sustentacular cells, another lineage from HBCs to microvillus cells via globose basal cells, and then the third lineage from HBCs to mature olfactory sensory neurons through GBCs. And it turns out that in the first lineage, sustentacular cells are produced via direct conversion of HBCs without cell division. By contrast, for the second and third lineage, we have a more aggressive, if you want, differentiation process where there's a proliferative state, the GBCs. It turns out that the ability to provide some supervision for the leaf nodes was quite important here, because without providing leaf node supervision, we would draw the false conclusion that sustentacular cells could develop into GBCs. So here is a diagram of the results. So our 3 lineages, so HBCs to sustentacular cells and HBCs to microvillus cells and olfactory sensory neurons via GBCs. This is our 3-dimensional representation of the expression data using principal chromosome analysis. Here's step 1 of Slingshot with the minimal spanning tree. And here's step 2 of Slingshot is fitting the simultaneous principal curves and then projecting onto the curves to get the pseudotimes. Any questions before I talk about TradeSeq? Okay, so Slingshot already provides quite a bit of information. So after running Slingshots, we have a trajectory structure, and we have for each cell pseudotimes and weights for each of the lineages. Now, the next natural question after having done trajectory inference is to find which genes are associated with the lineage in order to get an understanding of the biological processes that underlie differentiation. So most of the approach that had been proposed to date assessed differential expression between discrete cell clusters as opposed to exploiting the continuous resolution that we get from trajectory inference. So in a paper, Nature Communication paper that we worked on with Kelly Street, Hector Roudot-Bézieux, and Koen van den Berg, we proposed TradeSeq. It's a method that's based on generalized additive models based on the negative binomial distribution. And it allows to address 2 main questions. It allows us to study within lineage differential expression by looking for association between gene expression and pseudotimes over an entire lineage or portions of the lineage. And it also allows us to examine differential expression between lineages by comparing gene expression either over the entire lineages or specific points on pairs of lineages. And we can test for different types of D patterns by testing null hypotheses for linear combinations of the coefficients of the negative binomial generalized additive model using standard wald test statistics. And one nice output also of the trait-seq method is that we can use the fitted values to cluster genes. And the trait-seq method is also implemented in a bioconductor R package. So here's the NB-GAM underlying trait-seq. So this is just a standard generalized additive model with a negative binomial distribution. And here the key aspect of trait-seq is how we parameterize the mean. So the mean is parameterized in terms of smoothers that are gene specific, that's the G index, and lineage specific. Here are where the pseudotimes go in. And the Z's are functions of the weights that we get from slingshot. This is really the hard, these are just offsets for read counts. And these are experimental design variable. Really the key of the NB-GAM for trait-seq is right here. So we use cubic splines for the smoothers right here. And the tests in trait-seq will be based on the regression coefficients for the cubic spline. So these regression coefficients beta are gene specific, lineage specific, and not specific. So by looking at different contrasts of these regression coefficients, we'll be able to detect different types of gene expression patterns. So probably easier to look at a picture or a series of pictures. So this is for one particular gene. These are the kind of plots that we're going to be getting from trait-seq. This is a scatter plot of the log expression measure of the gene versus pseudotimes, and it's colored by the cellular states, GBCs, HBCs, olfactory sensory neuron, and so on. And these are the results of trait-seq from fitting the generalized additive model. We have one fitted curve here per lineage. You have the neuronal lineage in dark blue or purple here, the sustentacular lineage in teal, and the HBC in yellow right here. So here's a cartoon of the types of tests that we can do in trait-seq. So again, our usual plot for a particular gene, log count versus pseudotime. So trait-seq fits the GAM gene by gene. And here are the different types of tests with a toy model where we have two lineages. We have an orange lineage and a blue lineage. And the first two tests look at differential expression within a lineage, in this example, the orange lineage. And the last three tests look at differential expression between the orange and the blue lineage. So here, if we look within the orange lineage, there's differential expression in the sense that the expression measures for the genes are associated with the pseudotime. So this would be detected by an association test. There's also a difference between the start and the end of the lineage. So we would also detect this with the start versus end test. However, the blue and the orange lineages are identical. If we look at the second case study here, the orange lineage has no differential expression at all within it. However, it's different than the blue lineage. So we pick up differences with the between lineage test. Let's look here at the fourth case study. So within the orange lineage, we have D throughout the lineage, also between the start and end of the lineage. However, the orange and the blue lineage start at the same point and end at the same point. So the different end test for comparing the orange and blue lineage would not find differential expression. There are different patterns along the orange and blue lineages. This would be picked up by the pattern test. And there's also a difference in how the differentiation occurs early on in the two lineages. So this would be picked on by the early DE test. So instead of having just one test of differential expression for each gene, we could look at a variety of different types of expression pattern, both within and between lineages. So this is an overview of the functionality of trait-seq. Now we applied trait-seq to our olfactory epithelium data set. So these are the results here in panel A that we get from Slingshot. We have our three lineages that end up in the synapticular cell, the microvillus cells, and the neuronal cells. And here are some of the genes that were picked up as being differentially expressed by various tests of Slingshot. So this is in panel D. And then in panel C, we've clustered the top D genes based on the fitted values from the negative binomial GAM and revealing different types of clusters of the genes. So in this case, trait-seq helped us uncover the transcriptional programs that were underlying each of the lineages. It revealed both known and unknown marker genes associated with the lineages. When we look at a heat map of the top 200 D genes based on the association test, so if you remember the association test, that's a test that looks at DE within a lineage. So when we look at the heat map for the association test, the top 200 D genes, we get 5 gene cluster which have a different region of activity during the developmental process. So it's this cluster here. We also found 4 transcription factor discovered by the early D test that are involved in epithelial cell differentiation. So these were these 4 genes here that have very different expression patterns along the different lineages. And we were able to identify also a transcription factor activity cascade by looking for transcription factors that were significantly increasing over pseudotime, based on now we're looking not at the GAM, but at the first derivatives of the GAM. And it's in this diagram here. Any questions? And I'll close with the last 5 minutes on brand new work that builds on Slingshot and trait-seq. So far with Slingshot and trait-seq, we looked at trajectory inference for one condition at a time. We extended these framework to looking at multiple conditions. So studies that involve multiple conditions like wild type and knockout stem cell population often involve processes that can be described by a trajectory. So using the underlying trajectory structure could increase the interpretability of the result, as well as our ability to detect meaningful changes between conditions. So in our latest paper with Hector, Kuhn, and Kelly, we proposed the condiments package, which is a general workflow to analyze dynamic processes under multiple conditions by building on the trajectory structure. So this workflow not only addresses the inference of trajectories under multiple conditions, but it also allows us to compare trajectories between conditions in terms of a variety of biologically relevant properties. And we also have a biostructure R package for condiments. So there are 3 main steps in condiments. So the first step looks at different topology. In other words, suppose we have wild type cells and then knockout cells. The first question is, do we fit a common trajectory for these 2 cell populations, or do we fit a trajectory to the knockout and a trajectory to the wild type? The second question is about differential abundance. What are the global differences between conditions? So for example, do cells from different conditions behave similarly, either within or between lineages, as they progress along a common trajectory, or even common portions of condition-specific trajectories. So we looked at two types of variations within trajectories or portions of shared trajectories. So even when the two conditions have a shared trajectory or partially shared trajectory, the cells may progress at different rates within the lineages for different conditions. For example, the treatment might limit or speed up the differentiation of the cells. In this case, we would expect to have more treated cells compared to control cells at the early stages of the lineages. Another type of differential expression would be if the cells differentiate in varying proportion between the lineages for different conditions. So for example, the treatment might lead to preferential differentiation along one lineage over another compared to the control. So this is still like global changes between conditions at the level of trajectories or lineages. So the third step of the Kahneman's workflow looks at gene level differential expression. So the question here is, which genes have different expression patterns between the conditions? So for this purpose, we extended the trait-seq model, the negative binomial GAM, to allow condition-specific smoothers. And then we have a test, the condition test, that looks for differences in smoothers between condition using, again, wald test statistics and the regression coefficients. So here's a cartoon showing what Kahneman's does. This is a simple example where we have two branching lineages, lineage 1 and lineage 2. And here we have two cell populations, the wild types in blue and the knockouts in red. So in this first example, the knockouts and the wild type share the same trajectory topology, so two branching lineages. And also they share similar patterns. They're distributed similarly along the lineages of the trajectory. So we would say there's no differential topology, no differential progression, and no differential differentiation. In this example in panel C, we have the same topology, so we fit only one common trajectory. But if you look at lineage 2, there's differential progression of the cells. The blue cells migrate more quickly down lineage 2. In other words, it seems like the treatment blocks differentiation along lineage 2. In the example in panel D, we have the same topology again, but we also have differential progression in both lineages. There's more red cells, more treated cells towards the end of the two lineages. And there's also some amount of differential differentiation because of the speed up. So there's uneven distributions of the cells in the two lineages. In the example in panel E, there are different trajectories for the knockout cells here in red and the wild type cells in blue, but they still have the same topology, so we'll be able to map between the two trajectories. There's no differential progression and no differential differentiation. In the last example here, there's completely different topologies for the knockout and the wild type, so we would just reject the fit of a common trajectory, and there would be no way of doing further analyses for this example. So I think I'll have to stop here, but I'd be happy to answer any questions you may have. Thank you very much for your attention.
Thank you so much. Any question? So you can either send your question through the chat function on Zoom, or you can unmute yourself. Maybe I can ask sorry, go ahead.
Oh, yeah, I was just gonna, to get things started off, I had a question that I was interested in hearing your thoughts on. So a lot of some of the more recent methods in dimensionality reduction have worked on sort of smart methods for that. And I think ZINB Wave is a great example of that, or SCPCA. So methods that are doing dimensionality reduction in ways that account for technical noise, batch effects, other covariates. To what extent do you think this, or how would you weigh the pros and cons of that sort of approach versus a normalization and then dimensionality reduction approach?
Oh, that's a tough question. It's a really good question, but it's a tough question. I think there's all, the usual trade-offs that you have. One method that tries to do everything, right? You probably have more assumptions on that, and it's hard to check each of the individual steps. Whereas, I often feel more comfortable doing one step at a time because you can sort of check in between them. So that's what I would be more concerned about, is being able to check the assumption and validate both of them. It's probably not a very satisfactory answer, but I feel more comfortable having, little steps that I can check each time. Although I realize, like with ZinB-Wave, we tried to do everything at the same time, right? And it worked quite well, but I feel like they're more mysterious, a little bit more black box. Yeah.
Another question? Maybe I can ask a naive question. So for the input data right now, so does that work for the time course data, single cell RNA-seq data, or can you actually tell us a little bit more about to better use your tools, and what type of input data will be better to analyze the data and then better use your tool?
Oh, I see, right. So based on the experimental design. So I think the initial tools, like looking at exploratory analysis and normalization, could work for any type of data that you have. And the time course data question is very interesting. In that sense, you could, you wouldn't do really the lineage inference necessarily, but you could leverage trait-seq to do that, because you would have still these plots instead of having pseudo times that we have in trait-seq from the lineage inference. You could have the actual times and then use the negative binomial GAM to study differential expression associated with time. So you'd have chronological time in trait-seq as opposed to this kind of developmental transcriptional time. So you could certainly leverage trait-seq that way.
Great.
And I think, Kelly, did you guys do that? I think I vaguely remember you tried working with real chronological time with trait-seq.
I think Kuhn might have done that.
I think
we've done some like bulk RNA-seq with real time.
It was bulk. That's right.
Yeah.
Essentially, I mean, I lost the slides already, but the model is quite general, because the response that you have is your expression measure, right? And then you model the expression measure in terms of smooth functions of time and of experimental variable describing the samples that you've collected. So the smooth function of time, that the time we focused on was the pseudo time in trajectory inference, but you could put any time that you have. So you could start with a genuine time course data and fit these models and address the same types of questions. You would just skip the intermediate step of inferring the lineages. You wouldn't do the slingshot step, basically.
Got it.
Yeah.
Great. Thank you.
Yeah, that's a good question. Yeah.
Any other question? If you don't have any other question, thank you guys so much. Thank you, Sandrine.
Thank you all for your attention. I always try to present too much as usual.
Very interesting talk.
Thanks for having me over.
Yeah, if you have any other question, I think you're also welcome to contact Sandrine by emails.
Yeah, email's great. Yeah, please do. Thank you very much. Have a great day.
You too.
Bye.
Thank you. Bye.
Bye.